This video tutorial is about the structure of the mitochondria. Mitochondria are organelles that are present in all types of eukaryotic cells. Although mitochondria were first identified in animal cells in 1840 and in plant cells in 1900, their ultrastructure was not worked out until the 1950s, after extensive studies using electron microscopes. The mitochondria may be rod-shaped, thread-like, or spherical, with diameters usually of 0.5 to 1 micrometers and lengths of 2 to 5 micrometers, but occasionally they can be up to 10 micrometers in length. All mitochondria have an inner and outer phospholipid membrane making up the envelope. The outer membrane is smooth and the inner membrane is folded into cristae, or the singular being cristae, giving it a large surface area. Embedded in the inner membrane are proteins that transport electrons and protein channels associated with ATP synthase enzymes that allow protons to diffuse through them. Between the inner and outer mitochondrial membranes of the envelope is the intermembrane space. The mitochondrial matrix enclosed by the inner membrane is semi-rigid and gel-like. It contains mitochondrial ribosomes, looped mitochondrial DNA, and enzymes for the link reaction and Krebs cycle. So just thinking about the matrix then. The matrix is where the link reaction and the Krebs cycle take place. It contains enzymes that catalyze the stages of these reactions, molecules of the coenzymes NAD and FAD, oxyl acetate, which is the 4-carbon compound that accepts the acetyl group from the link reaction, mitochondrial DNA, some of which codes for the mitochondrial enzymes and other proteins, as well as mitochondrial ribosomes, which are structurally similar to prokaryotic ribosomes, where the proteins are assembled. Next, just thinking about the outer membrane. So the phospholipid bilayer composition of the outer membrane is similar to that of membranes around other organelles in eukaryotic cells. It contains proteins, some of which form channels or carriers that allow passage of molecules such as pyruvate into the mitochondrion. Then the inner membrane, the lipid composition of the inner membrane differs from that of the outer membrane. The lipid bilayer is less permeable to small ions, such as hydrogen ions or protons, than the outer membrane is. The folds, or the cristae, in the inner membrane give a large surface area for the electron carriers and ATP synthase enzymes embedded in them. The electron carriers are protein complexes arranged in electron transport chains. Electron transport chains are involved in the final stage of aerobic respiration, which is oxidative phosphorylation. Next, we're just going to mention the intermembrane space. So the intermembrane space between the outer and inner layers of the mitochondrial envelope is also involved in oxidative phosphorylation. The inner membrane is in close contact with the mitochondrial matrix, so the molecules of reduced NAD and FAD can easily deliver hydrogens to the electron transport chain. Thinking then about the electron transport chain, this can sort of shown on the slide in front of you and again on the slide that is now in front of you. We see each electron carrier protein contains a cofactor, a non-protein heme group that contains an iron. The iron can accept and donate electrons because it can become reduced, so Fe2+, by gaining an electron, and then it can become oxidized, which would enable it to be Fe3+, when donating the electron to the next electron carrier. Electron carrier proteins are oxidoreductase enzymes. The electron carriers also have a coenzyme that, using energy released from the enzymes, pumps protons from the matrix into the intermembrane space. Protons accumulate in the intermembrane space and a proton gradient forms across the membrane. The proton gradient can produce a flow of protons through the channels in the ATP synthase enzymes to make ATP. You'll learn more about this in another topic, and hopefully you have already learned about the synthesis of ATP by phosphorylation. Just a final word then on this ATP synthase enzyme. The ATP synthase enzyme is a large enzyme that protrudes from the inner membrane into the matrix. Protons can pass through them, and it's shown on the slide in front of you.